Imagine spending months or years in space. Your blood floats weightlessly in your body. You have little protection from cosmic rays hurtling through space. What will that experience do to astronauts traveling to Mars? How can scientists keep them safe?
First, you need to know what's changed or what the risk areas are before you can actually try and prevent them or correct them. You break open the cells.
Chris Mason is part of a scientific team that's investigating life in space, all the way down to the level of DNA.
This is the first step on a ladder towards understanding what could happen to missions to Mars for human physiology.
And liftoff. A year in space starts now.
In March 2015, astronaut Scott Kelly began a year-long orbit of Earth aboard the International Space Station. One mission objective was to see what a year in space does to the activity of an astronaut's genes.
This is really a privilege to be able to do this. And
NASA picked out Kelly for the study because he has an identical twin. Mark Kelly is an astronaut too, but he spent the year back on Earth.
I told NASA, whatever you need, just tell me where to be and when, and I'll give you whatever you want.
Two people with an identical genome in two very different environments.
there are things from a genetic perspective that you can learn from using him as a control and me as an on-orbit human subject.
This is the lab, mostly. I'll give you the quick tour.
Chris and his colleagues set up a baseline for their study by analyzing samples from the Kelly brothers 6 months before the mission. Their plan was to compare those baseline results to the samples they got all throughout the mission, and then 6 months after Scott returned to Earth.
So you're going to be putting in individual astronaut cells and then looking inside each one.
The samples include blood, saliva swabs, and fecal material. While at the space station, astronaut Kelly collected his own samples and sent them back down to Earth.
One of the more exciting times was getting the fresh blood draw from space. And
so fresh blood from space.
Yeah,
that must have been...
It was very exciting. I felt like, not quite like being visited by aliens, but it was, almost as exciting maybe.
Since no one had done this before, you must have been a little worried, is it going to be any good?
Cells seem surprisingly tolerant to being thrown around the atmosphere and the planet.
We, put them for long-term storage into the freezer, and in here, this is where they all basically get kept. There was one day when we couldn't actually find this box. There was only about 30 minutes where it was not found, but it was a terrifying 30 minutes.
Did everything work out without a hitch, or did you get some problems along the way?
So some of the challenges were technical, just getting samples up to space and back. One of the SpaceX resupply missions exploded on the way up to the space station. So we lost some of our supplies, and actually we got this really nice letter from the director of NASA saying, space is hard, but hang in there, we're going to send another rocket soon.
One of the most important things that Chris and his colleagues are studying is the way that molecules in our cells control our genes. This field is sometimes called epigenetics. Our genes are surrounded by other molecules that can turn them on or keep them silenced. If those molecules fall off our DNA or change in some other way, our genes may start acting differently.
What's extraordinary is you can almost perfectly predict someone's age just from their epigenome. So we'll be able to see where they're differing and how fast has that difference changed because of space travel.
Is there a reason to think that going up into space for a long time might have an effect on aging?
So we hypothesized that the stress of space travel will age him slightly more than his identical twin on Earth. So even though technically from Einstein's relativity he's a little bit younger, the stress of space travel may age him in other ways.
Scott Kelly back on Mother Earth after 340 days in space.
So now you're getting all this material. What's the plan? How long is it going to take you to work through all the data and see anything?
As soon as the last samples are collected, we'll extract and sequence and characterize all the molecular changes. So very soon we'll be able to get a sense of what is the real genetic and epigenetic risk for astronauts.
This mission is the latest achievement in our country's space program, but it's not the last. It's in our DNA of our country to explore, and we must never stop doing this.